Hey there and welcome. Have you ever, taken a pill for something and just stopped to wonder, how does it actually work? It seems like magic, right? Well, today we're going to pull back the curtain and take a pretty wild journey deep inside our own cells to figure out the science of how medicines get the job done. Let's get right into it. So, here's a question I bet you've asked yourself. How does a pill you take for a headache know to go to your head? Right? You swallow it and poof, the pain's gone. Is it some kind of tiny, smart missile that just hones in on the problem? Well, the truth, it's way more interesting than that. Okay, so to get our heads around this, you've got to think about your body as this huge, incredibly complex communication network. I mean, it's constantly sending signals everywhere. And drugs, they're basically all about hacking into that system and sending the right messages to the right places.
And here's the big secret. That headache pill, it doesn't know where to go at all. Nope. Once you swallow it, that drug goes on a tour of your entire body through your bloodstream. The real magic isn't about the journey, it's about the destination. It only acts on the specific little targets that are causing your pain. But, and this is key, those same targets can exist in other parts of your body too.
There's actually a fancy name for this whole field of study, pharmacodynamics. It sounds complicated, but really it's just the science of what a drug does to the body. It's all about what happens once that drug molecule arrives at its destination. So what exactly are these destinations we're talking about? So drugs are looking for a place to land, and they've got four main options, four types of proteins they can interact with. You've got enzymes, ion channels, carrier proteins. They're all super important. But the big one, the one that the vast majority of drugs are designed to hit, that's receptors. Think of them as the master control switches for our cells. They're the most powerful targets we have, so yeah, they're going to be the star of the show today. And here's the best way to picture this, an analogy we're going to use a lot. Imagine every single one of your cells is covered in thousands of tiny, very specific locks. Those are your receptors. And a drug is just a specially designed key. And just like with real locks and keys, only the right key can fit into a specific lock and make something happen.
All right, let's check out our first type of lock. We'll start with the simplest, most straightforward ones around, ligand-gated ion channels. You can think of these as the really fast-acting gatekeepers of the cell. The whole lock and key thing is perfect here. The key is just a molecule. In the science world, we call it a ligand. When that key fits into the lock, the receptor, bam! The gate swings open instantly. It's a direct physical action, and it happens in the blink of an eye, literally, in milliseconds.
So, the process is dead simple. The key binds, the gate opens, and a flood of ions rushes into the cell. What's so important about that? Well, it instantly changes the cell's electrical charge. This is a huge deal, especially in our brain. It's how we turn chemical signals into electrical signals just like that.
But, of course, not all locks are that simple. Let's move on to a much more common and, frankly, a much more sophisticated type of lock. These guys act less like a gate and more like a signal dispatcher. They're called G-protein-coupled receptors, or just GPCRs for short.
And the analogy changes here, which really shows the difference. With a GPCR, the key doesn't open the door. Instead, it's like ringing a doorbell. The key binding to the outside of the cell rings the bell, which then activates a messenger on the inside to get up and go do a bunch of different jobs.
And just to show you how ridiculously important these doorbells are in medicine, get this. About 36%, that's more than a third, of all drugs approved by the FDA work by targeting these GPCRs. That's huge. It makes them one of the absolute biggest targets for new drug development.
So you can see the process is a bit more involved here, right? The key binds. This makes the receptor literally change its shape. That shape change wakes up a messenger, the G protein, on the inside, and that messenger then sets off a whole chain reaction of events inside the cell. It's a slower process than just opening a gate, for sure, but the signal can be much, much bigger.
Okay, so we've got these locks all over our cells. Now for the really cool part. How do scientists actually design the keys, the drugs, to hack the system for our benefit? This is where we talk about the difference between turning a system on and slamming it shut.
So you can basically design two kinds of keys. First, you have an agonist. This is like the perfect master key. It fits the lock, it turns, and it opens the door, activating the cell's response. Then you have the antagonist. This key is a little different. It fits in the lock perfectly, but it doesn't turn. It just gets stuck in there, jamming the lock and blocking any other keys, including your body's own natural keys, from getting in and opening it.
But wait, it gets even cooler. It's not just a simple on or off switch. Scientists can design keys that have a whole range of effects. A full agonist is like turning the volume to 10. You get the maximum response. A partial agonist turns the volume to, say, a 3 or a 4. It works, but the response is weaker. And then you have these really clever ones called inverse agonists. They actually turn the volume down from its normal resting state, which is perfect for calming down systems that are a little too hyperactive.
So this level of precision is just incredible, right? But it also brings us face to face with the biggest challenge in making medicine: side effects. So the question is, how can everything we're learning about these locks and keys help us build smarter and maybe even safer drugs in the future?
Let's go right back to our lock and key analogy. The problem of side effects, a lot of the time, is that our key isn't perfectly specific. What if the key we made to stop your headache also happens to fit, maybe not perfectly, but well enough, into a few other locks somewhere else in your body? You know, the lock that controls drowsiness or the one that controls your stomach. Well, when it opens those locks too, boom, that's a side effect.
And this is where we get to the absolute cutting edge of pharmacology. It's a concept called biased agonism. Okay, remember how we said the GPCR doorbell sends a messenger inside? Well, it turns out one doorbell can actually send out multiple messengers, each with a different job. What biased agonism is all about is designing a key that rings that doorbell in such a precise way that it only activates the messenger for the good effect we want and completely ignores the other messengers that cause all the nasty side effects.
I mean, the goal here is nothing short of revolutionary. Picture a really complex lock. It's got two sets of tumblers inside. One set triggers the good thing you want, the pain relief. The other set triggers the bad thing, the side effect. A biased agonist is like a key that's been cut with such insane precision that it only turns the good set of tumblers and it leaves the side effect tumblers completely untouched. And all of this leads us to one huge final question. With this kind of mind-blowing precision on the horizon, could we one day design medicines with zero side effects? Medicines that do exactly what we need them to do and absolutely nothing else? Well, the science of biased agonism says that future might be a lot closer than you think. It really could be the dawn of a whole new era of smarter, safer drugs for every single one of us.